to how many of you are actually using genomics right now in your practice? So a good percentage, and that's great. Because this is our foundations half day, I am going to review some things, so there will be review for those of you who are using it, but I think everyone will find some new information here. Let's see. As she said, I'm the co-founder and medical director of Intelix DNA, so that's and I'm not going to go over what the goals are and the formal learning objectives, because we're just going to jump right in to a little easier form of the goals. A good friend of mine said, when you're teaching, you want them to know, you want to think about what do you want them to know, what do you want them to think, and what do you want them to do at the end of the talk. And so what I'm hoping is that at the end of this talk, you're going to really feel comfortable explaining to your patients the difference between genetics and genomics, that you're going to think that using genomics in your practice is really exciting and something that will add to your ability for clinical decisions, and that you will start doing genomics consults or expanding what you're doing with genomics. Let's start with the fundamentals. What's the difference between a genetic disease and genomics? Well, genetic diseases tend to be caused by fairly large genetic events. So think about trisomy 21. You have a duplication of a chromosome. Think about Huntington's disease. You get multiple extra repeats of a segment of DNA. Think about deletions like Turner syndrome. Those are big genetic events. Now, sometimes you can have a pathological disease because of a genomic event. There are many different kinds of Tay-Sachs, and over 30 of the types of Tay-Sachs are caused by a genomic event, a single change in a polymorphism. Those are considered pathogenic polymorphisms, what ClinVar has listed as pathogenic. That's not what you guys are going to be focusing on. That's not what I focus on as a family physician or integrative medicine physician. I focus on the ones, the SNPs that can change function, that I can modify, that I can help my patients by knowing about. So what's genomics? Genomics refers to those small genetic events, generally a one nucleotide, or as I explain it to patients, a one-letter change in DNA, or a very small insertion or deletion, a 1 or 2 or 3 nucleotide base pairs. And these create changes in the DNA, which then create changes in the proteins expressed that can lead to less or non-functional genes, or less or more of those different proteins or enzymes or receptors or whatever it's coding for. Let me step back a little bit just for the people who are new or who it's been decades since they took a genetics class. Remember, you've got DNA and chromosomes. So you've got your chromosomes, which are made up of bundles of DNA wound around histones, and that these chromosomes carry your genetic information. That all the information together is your genome. Of course, you have the DNA, which is the double-stranded helix composed of the base pairs and the sugars. And so the gene is one unit of DNA that carries instructions for making a specific protein or receptor or vitamin or whatever it's coding for, or a set of genes causing that. And that together, these really determine not just heredity, but who you are. Your genome is your complete set of DNA. You have about 20 to 25,000 genes in your genome, but that doesn't mean that there's only 20 to 25,000 SNPs. Let's go a step further. What's an allele? So an allele is one or more of the alternate forms. You have 1 or 2 copies of your genes, of various forms of them, that you inherit for a specific form of a mutation or variant found on the same place in the chromosome. So this is getting closer to SNPs, because the allele, the difference between blue eyes and brown eyes is a SNP. So the major allele, the reason I want you to know the term allele is you're going to hear used major and minor allele. And the major allele is the one found in more than 50% of the population, and the minor is the one found in the smaller proportion. Now, there are minor variants or minor alleles that are only in 0.1% of the population or 2% of the population, and there are minor alleles where you're going to find them in 49% of the population. And so kind of knowing a little bit about, first of all, how prevalent a gene is may help you when you're talking to a patient, or how prevalent a SNP is, because take MTHFR, for example. Yes, it's a very important SNP, but you have to realize that in a heterozygous form, you're talking 45% of the population. So you don't want people to think they're dying because they have this, because if 45% of the population has it, you don't want them to think that they're a mutant. So try to use the word variants or SNPs and not mutations, because again, it's more like blue eyes and brown eyes. It's what makes us unique. However, if we understand what their variants are, we with nutrition, with supplementation, sometimes medications, lifestyle changes, can help support them for what they make more or less of. So, so the minor allele is, by definition, the variant. You'll also, of course, hear the terms homozygous and heterozygous. Homogeneous for the same, so homozygous means they have two of the same allele, and heterozygous means they have one copy of one and one copy of another. The base pairs are the building blocks of the DNA, that A, T, C, and G, the guanine, cytosine, adenine, and thymine. So now we get to a SNP. What are all these SNPs we're talking about? When I talk about it with my patients, because again, if this is new for you, think about it for patients. I say, I know it sounds like it should be a little piece of cloth, but what it is a little change in your DNA, and it actually stands for single nucleotide polymorphism, a one-letter change. I don't usually go into how DNA is made up of 4 letters. That's all they need to understand, a tiny little change that may or may not change the function of that gene. The difference between a SNP, the major and minor allele, is the SNP. So remember, you've got about 3 million SNPs or variants in the human genome, but that doesn't mean that every single one of them matters. You can have a change in your DNA that's in a non-coding section that has no change in it. You can change different nucleotides and it may slightly change the bending or the shape of a protein and not have any effect. Or you can have ones that are in a promoter or change to a stop codon that can cause a completely different expression of that gene. So the way I explain that is when we change one little letter of the DNA, sometimes it doesn't matter at all. It's kind of like changing the word from stir to blend in a recipe. Really doesn't make a difference. But other times, if you change bake to broil, it can be a big deal. So what we want to learn is which of these SNPs really make a big deal. You don't want to just say, oh, look, I have all of these various SNPs in the VDR pathway or the MTHFR pathway or BRCA or whatever pathway you're looking at in whatever product or whatever translation, because that's going to lump together the SNPs that are baked to broil with the ones that are blend to stir. So whatever you're doing with genomics, you have to know that if you're counseling someone about a particular SNP, that there is evidence that that SNP actually has some kind of an effect clinically. And that's probably one of the most important things that you can use moving forward with your genomics, is make sure that you know that what you're talking about with a SNP is a clinically significant SNP. So the central dogma of genomics is that DNA gets translated to mRNA, so, and the RNA gets translated to the proteins, and that if you have a little change in the DNA, it may change that RNA sequence, which may change the protein expression. The RNA, of course, the DNA is also in codons, but the RNA, of course, are in codons, set of 3 base pairs, and some of the reasons why it doesn't always change is, as you can see, under like valine and alanine and many of them, there's 2 or 3 of the 3-letter combinations that still make it be the same amino acid. I mean, that same, so that it really doesn't matter. You could change it and you're changing one alanine to another. So that's why not all SNPs matter. And again, just also how they affect the protein foldings and such. Let's talk about naming. There's actually one mistake on here. There's a couple of different naming conventions used just to make it crazy when you're going to the literature, because you may see different genes or SNPs referred to by RS number. That is the easiest. RS is a reference SNP, and that terminology really didn't start to get used until I find more like 2011. Most of the literature starts to switch over to that, and it stands for reference SNP, and that's great because it allows a lab over in Europe to translate and talk to a lab in Australia and America because they're all using the same number. But there's also positional naming, and MTHFR C677T should be a positional naming. The C gets changed to T. And then you can also do it with amino acid naming, MTHFR AL222 Val. So if you're going back to the literature on your own, which once in a while you may want to, don't get confused, or if you do get confused, we always say genomics is hard. Just know that all of us have been there. But just know that you're going to see these different terms and they all mean the same thing. So you get the gene. So now what do we do with the gene names? So the gene names often relate to function, but I don't want you to think that they always relate to function. There is a gene that increases the risk of melanoma that's called the cleft lip and palate gene, and it has nothing to do with the cleft lip. So it was just poorly named because they thought it was going to have something to do with it, and the name didn't get changed. But lots of times the gene will relate to function. The RS number is, again, that's very useful for international identification. And then there's the whole gene that can have multiple SNPs within. There could be 30, 40, 100 different MTHFR genes. Not all of them, SNPs, not all of them are significant. So the MTHFR C677T also can be seen as, instead of a T, an A. So if you're looking at a particular interpretation product and you're like, wait, this is supposed to be that famous MTHFR gene, why is my patient having A? Remember, T pairs with A and C pairs with G, and there's forward and reverse strand. Most of the products nowadays, the interpretation products, most of them are reporting on the forward strand, but a lot of the literature was done on the reverse strand. So MTHFR C677T, you're going to see A as the variant. Okay, so then just to make things more confusing, but please don't get up and leave, I promise you're going to get it, there's also triallelic SNPs. So sometimes, like there's a SNP in the complement pathway, which is important in macular degeneration, where there's 3 choices. There's an A, a G, or a T. And so again, you just have to kind of know these terms so that when you're reading about things, you're like, oh, I know what that means. And don't worry too much about it other than that. There's also insertions or deletions. How many of you guys have heard of the ACE gene, the ACE insertion? That's an insertion. What about ApoC1, which is found in, it's on the same gene as ApoE4 and affects cognition. If you haven't heard of that one, that also, again, is an insertion versus a deletion. Now, these aren't big deletions like in Turner syndrome. These are little 2 or 3 base pairs deletions. So you may, in some different genomic interpretation tools, see them reported as I's or D's. Now, just to be actually clear, I and D aren't the opposite. It's the insertion or the absence of an insertion, or a deletion or the absence of a deletion. But it's hard sometimes to figure out how to report things. Let's go back to gene names. Again, gene names can relate to function, but you guys want to really make sure you're getting accurate information about what a gene function is, because there's a lot of urban myth. And it wasn't for malice that this got propagated. It was because genomics is hard. But the VDR gene, for example, is a vitamin D receptor, but it's not the vitamin D receptor that relates to 125-OH vitamin D levels that carries vitamin D in the blood of 25-OH vitamin D. That relates to 25-OH. It's a 125-OH receptor. So it really does not relate heavily to vitamin D levels. There are other genes that relate to the synthesis of vitamin D and the first step of hydroxylation of vitamin D that are much, much more highly associated with vitamin D levels. And VDR relate because vitamin D receptors are really transcriptional factors. VDR affects things like osteoporosis, immune function, cardiac, all kinds of different things. And so it's important for you guys to kind of know and don't just kind of go, oh, it's called the VDR. I heard somewhere in a direct-to-consumer product that's related to vitamin D levels. Check out and learn academically, and that's why ANA is offering these kinds of courses so you can learn what things actually are. COMPTIDO, catecholamine, methyltransferase, the NOS, MTHFR, many genes you'll be able to figure out what they do by their names, but not always. SNPs in pathology. Sometimes a single SNP will have a significant effect. Factor V Leiden, for those of you who know that, it's a clotting factor, has implications just having one copy. But in other cases, you have pathology more or risk, health risk more because of the combination. So cardiac, we're going to look at, obesity, we're going to look at, blood sugar. And it's not one gene, it's really hundreds of genes, but then there may be 10 or 15 or 20 that are kind of big contributing factors. Health risks are also very much affected by epigenetics, and we're going to talk about what the classic definition of epigenetics is, but also nutrigenomics, which is kind of loosely a subset of epigenetics, where your DNA expression can be modified by what you eat and what you take into your body and supplements and such as well. Okay. The other thing that Dana touched upon that's important when you are counseling patients, you can't imagine that every single variant that a person has inherited is bad. That wouldn't make sense. So if you're talking to people about their DNA, don't only focus on variants that convey risk. There are also variants that convey benefit, that improve their vitamin D levels, that improve their immune system, that give them protection against all kinds of different things. So for example, in celiac disease, the IL-2-21 actually decreases the risk. So there's lots and lots of these examples. There's SNPs in platelet pathways that make you have less sticky platelets, almost like built-in aspirin, and decrease your cardiac risk. Another terminology that you guys have probably heard of, is because I'm sure all of you have dealt with celiac, is haplotypes. So haplotypes refers to a set of SNPs or a combination of alleles found on the same gene that tend to be inherited together. Not always inherited together, but their effects are more pronounced when they are. And when you hear about those HLA types that convey increased risk of celiac disease, so 2.5 is the most common, and 90% of celiacs have an HLA type of 2.5, which has like 3 other names. That's actually not one SNP, it's a combination, it's a haplotype, 2.2, all those things. Okay, so now let's kind of switch to things that are a little more fun, some case studies, and epigenetics and nutrigenomics. So epigenetics literally means on top of genetics. And the term epigenetics was first coined in 1942. Really crazy to think about that. And it kind of just laid dormant. But it had to do with a professor who was realizing that not all genes were expressed, and trying to understand that phenomenon. It didn't always correlate to a phenotype. And so epigenetics has really transformed the way we think about genomics. And so I'm going to use the classic example of epigenetics of the stress that your mother was under during pregnancy affects your risk of depression. That's actually a really amazing phenomenon. How does that work? Well, if you're talking to a geneticist or a PhD about epigenetics, they're going to say epigenetics is methylation. Methylation of CPG islands, and it's turning off the DNA. So that's the biggest category of epigenetics, is that our DNA is not all turned on, and methylation is very, very important for turning off the genes. You have tumor suppressor genes, you have all kinds of genes. Throughout the genome, genes can be turned on and off. And so proper methylation is how we keep genes from being overexpressed. Stress causes release of cortisol, and cortisol causes higher levels of glucocorticoid receptors to be expressed, and also lowers methylation of a host of genes. And lowered methylation is associated, again, with effects not only in the person having the lower effects, but generations to come, even up to the grandchildren. Now, MTHFR is an interesting gene because it has a twofold effect on depression. One is MTHFR is a big factor in methylation. You're going to see it come up in toxicity issues and all kinds of issues with methylation, cancer issues, lots of stuff. But MTHFR, as we'll talk about at other times, also is important for making norepinephrine, serotonin, and dopamine. So it kind of gets a double whammy for depression. So you can get this very nice little graph here where you can see that in black, the individuals who have 2 copies of the MTHFR C677T gene have a much different response. They're more prone to depression. They have a lower Beck depression inventory score than people who are better at methylation, because each MTHFR C677T gene decreases your MTHFR pathway about 30–35%. What about nutrigenomics? Well, you're going to find that nutrigenomics overlaps with methylation as well, and that's why I started with methylation as an example, because nutrigenomics is the scientific study of the interaction of nutrition and genes, especially with regards to the prevention and treatment of disease. But the reason it's a subset of epigenetics is nutrition. Things like choline, betaine, all of those things contribute to you having enough methyl donors. So, it makes sense that what we eat affects how our genes work. And so what you get from your food, from supplements, can affect epigenetics as well. Now nutrigenomics has become a wider field, and even though it's a subset of epigenetics, we don't just focus on methylation, because we know that food can affect inflammatory pathways. It can affect, obviously, vitamin levels, so many things. And so nutrigenomics, in the broad sense that you guys will all be using it and are using it, is how can you affect or respond to the genome and help optimize health in a personalized way by understanding what SNPs somebody has. I'm going to give the example of the agouti mouse. And I'm sure all of you guys at various times have heard of the agouti mouse. It's a classic epigenetic example. And the overactive agouti gene leads to decreased satiety via a hypothalamic receptor that is supposed to make you feel full, called MC4R. And so, of course, ghrelin, growl, your stomach makes you feel hungry. Ghrelin makes you hungry and leptin makes you feel full. So just remember grr for growl. So ghrelin stimulates the release of the agouti peptides that binds to the agouti receptor, and that turns off that satiety gene. So if you have a gene that's overactive, then you have overactive agouti signaling, and you're going to have, if you have a defect there, you're not going to turn it off, and you're going to stay hungry. So that big fat orange mouse is an agouti mouse. The interesting thing is, they're both agouti mice. How can that be? Well, they were fed different diets. That's why it's such an exciting opening pathway to talk about, because the obese mouse was fed a diet that was choline, folate, and betaine deficient. And choline, folate, and betaine are very, very important methyl donors. So it just goes to show you, on a very basic level, even knowing that what foods your patient needs, or if they can't get it through diet, to get enough choline and folate and betaine is really important. And what you're going to find as you delve deeper into genomics, there are certain people, like if you have problems in the choline synthesis pathway, which is PEMT, along with MTHFR, so that whole cycle, instead of needing 500 to 900 milligrams of choline as a typical amount, you need 1,000 to 2,000. So it really shifts it. So really food is ultimately what, of course, feeds our DNA, not things in a bottle. But sometimes you're going to find the needs are so high that you can't work it all into someone's diet. What about our environment? I consider, because I'm not a PhD, so I go a little more liberal, I consider environmental effects on genes part of epigenetics, because we don't have a different word for it. I'm sure we'll invent one eventually. But BPA turns on the agouti pathway and can trigger weight gain. So the BPA from plastics, so I'm so thankful to all the different Yeti and stainless steel cups, until we find out what stainless steel does, that we're not constantly throwing water bottles around, because the amount of plastics that get into our system that reduce methylation and affect all these different gene expressions have been associated with all kinds of things, such as adult onset diabetes, tumorigenesis, obesity. And BPA is still in 16,000 foods on American shelves because it has not been outlawed here because so many canned products, the inside of jars, there's still a long way to go, but it's getting better. So let's talk about using genomics to target dietary supplement recommendations, because at the end of the day, that's what you guys want to be able to do, is to sit down with a client, with a patient, and say, we've looked at your genes, we've looked at your genomics, you're having these symptoms, so you don't want to use genomics in a vacuum. You're going to use it as a clinical decision tool along with their history, because somebody who's having fatigue or cognitive impairment is different than how you're going to respond to somebody who's just there for prevention, kind of to learn for fun. So let's start with obesity, since we were just talking a little about that. Before we look more at obesity SNPs, I want to take you back 1,000 years, because we think of obesity as a risk, but 1,000 years ago, these SNPs were benefits. If you lived in Eastern Europe during a time of famines, and you knew that this winter was a good chance you were going to have to slaughter that cow, and now it's been the beginning of winter and you can't afford to feed the cow anymore, so you're slaughtering it, and you don't know when you're next going to have any good animal protein or protein access, it's not in your favor to kind of go, oh, I'm hungry, I'll just want a little bit, thank you. You want to be hungry and eat a lot of it, because that's what's going to get you through the winter. But now you can go get saturated fat every single day of your life. So SNPs that made you turn off your satiety in response to saturated fat are a risk factor for obesity, but they are a benefit for starvation. So leptin receptors, that MC4R, which is that hypothalamic gene that causes you to not feel satiated, all are turned off and affected by saturated fat. Interestingly, it only takes about 12 grams a day of saturated fat or more, which is not very much if you look at, you guys know, again, know that better than I, to affect those genes. And so a high-fat diet, it really should say high saturated fat, because these genes are not affected by the monounsaturated fats, by the omega-3s, things like that. So it's not about the nuts and the seeds and the olive oil and the fish. It's the saturated fat can have a huge effect on the wide variety of the leptin, again, leptin receptors, MC4R, I call the snacking gene because it affects satiety, but also high fructose corn syrup also triggers leptin resistance and can stimulate hyperphagia. And as you guys, again, already know, there's high fructose corn syrup in everything on all these commercial products. You can have it in your ketchup. I want to spend a moment on STAT3, because STAT3 is a really great example of where, if you know their STAT3 status of the STAT3 SNPs that have been shown to be significant, you can really help your patient with their waistline. So STAT3 is involved both in leptin signaling and inflammation, and we all know that fat around the middle is inflammatory, and it's associated particularly with abdominal obesity. And what you'll see is the blue line on the bottom is individuals who, of the 7 potential, because the first ones are, the first 3 are significant even in 1 copy, and then the bottom in the 2 copies. So of the 7 potential SNPs that have been associated with the STAT3 abdominal obesity, if you have less than 2, doesn't really matter if you increase the amount of saturated fat in your diet, not much change in your waistline. But you are somebody who has 3 or 4 or 5, then the more saturated fat that you eat, the direct proportional logarithmic increase in abdominal obesity. And again, it may have been a while since you did logarithmic math, but we all know that that's like that. And so if somebody knows that they are at high risk because of STAT3, all they need to do, STAT3 is highly sensitive to saturated fat, but not unsaturated. And if you can see on the graph, it's a little hard to see, the lines intersect at about 8% of your calories from more than saturated fat. So in order with someone who's a STAT3 person to keep them from gaining weight, you've got to teach them they can still have fat in their diet, but they've really, really got to limit their saturated fat, which generally means getting rid of, for the most part, red meat, cheese, lard, bacon, and even coconut oil, of course, is highly saturated. This is from a patient of mine who has, and I'm not showing you all the obesity genes because, again, we'd have to have 2 hours on any one topic, but one of the things she had was a lot of STAT3. And when we switched her to a mostly plant-based diet and fish, which is the best way to describe a low-sat-fat diet, she went from a size 12, and now she runs between a 6 or an 8, and it's been well over a year. And she doesn't count calories or do anything like that. She just switched how she viewed food and what was good for her. But again, the leptin receptors on those are about a less than 12 grams a day is ideal, but the most sensitive to saturated fat, less than 10% sat fat, which is a very low saturated fat, are people with the STAT3. Most of my examples are all Nutrigenomics, kind of dietitian, nutritionist common issues, but I did want to put a little bit of an example of cardiac because you're going to find, of course, which you guys already know, nutrition affects everything. So let's look at cardiac genomics, a few genes for a moment. 25 to 35% of Americans die from a cardiac-related death. And we all know that cholesterol just explains a tiny little bit of it. That is not the main cause of cardiac illness. So here's a patient of mine, a 62-year-old active male. And this is actually not him. This guy looks like a BioT commercial, you know, testosterone buff guy. But he's kind of a lean, slender runner. And he's been really trying to do everything right. I mean, he has a strong family history of heart disease. His brother had something at 37, had his first stent. So he really tried to be very healthy. He was running, eating well, but had such a strong family history that when I saw him now about 1.5 years ago for his annual, he told me he was only running. I said, "Are you still running?" And he goes, "Yeah, but only 3 miles." I'm like, "Why only 3 miles?" Because I knew he used to run 5, 6, 7 miles at a time at least. And he goes, "Well, about mile 3, my left shoulder starts to hurt." I'm like, okay. So we sent him to the cardiologist. Turns out he had a left main that was pretty blocked, that's called the widowmaker. That's a big deal for those of you who don't know the different arteries. Had triple bypass. And so then he comes back to me, okay, what can we do to decrease my risk of getting any further arteries blocked? Because, there's only so many that you have. And so this is part of his genomics. Again, there's many more pathways to this, but I want to show you examples of 2 of the pathways that we then addressed. So 2 of the pathways that he had one called the CDKN2A. Don't worry about the names. If you start doing genomics, the best thing that you can do for yourself is not feel that there's anything called oversimplification. So I come up with a term that's so easy. I say, that's the calcification pathway. Because to explain all the ways that a gene can cause arterial stiffness, you're going to have the person across from you asleep. So you need one-liners. So the CDK is the calcification pathway. So I wanted to address that with him. And then he has a number of inflammatory pathways. I'm showing you one of them. He actually has 3 or 4 other inflammatory pathways that also have been highly associated. And this particular one works in combination, and he has both parts of the combination. So what do we do about that? Before I go on, I wanted to, as an aside, do you see how there's 2 genes here? And you might look at something and see, oh, someone has a bunch of SNPs. Many times, SNPs are highly linked, meaning it's called linkage disequilibrium. It actually seems like it should be called linkage equilibrium, not dis, because it works together. You'll often see that some SNPs are frequently co-inherited. So these 2 particular SNPs, even though he has 2 copies of both of them, over 90% of the time, you inherit both. So just kind of, if you hear that term, that's what it means. So anyway, what did we do about his calcification? Well, calcification of the arteries is tremendously affected by your vitamin K2 levels. And these are actual aortic biopsies that were done over in the Netherlands. And you can see behind the biopsies these faint gray lines. And on the healthy aorta, you can see that the tissue levels of vitamin K2 were much higher than in the calcified arteries. And what we know is vitamin K2 helps to direct the calcium to go to the bones and not to the heart. And again, if you want to know the pathways, it's matrix, GLA, and then there's also the OPG pathway for the bones, but I wouldn't overly worry about it. Just kind of, again, I explain it to the patients. Vitamin K2 helps the calcium to go to the bones, not to the heart. And you have a gene that's very common, it's again almost half the population, that makes you more prone to putting calcium in your arteries. And I say, it's almost half the population, but the reason I'm talking to you about it is because it's really easy to address. And so how do we address it? And I remember, heart disease is common too. So a lot of the genes that are variants that have significant effects in things like diabetes, heart disease, osteoporosis, inflammation, many of them will be common because those are common things that you see. So how do we address it? We address it with vitamin K2. It doesn't take a huge amount of vitamin K2 to make a difference. This was a study showing, and it was 4,800 patients for 10 years, so a good-sized study for a nutritional study, that if you had your vitamin K2 levels less than 21 micrograms a day compared to 32 or more micrograms a day, that translated into an over 50% reduction in both cardiac and overall mortality. Now, that's huge. Now, knowing that that's the case and that more than 50% of people have the vitamin K2, these calcification SNPs that can benefit from it, the logical thing would be for us to have a national campaign to make sure everybody gets enough vitamin K2 in their diet, but it's not going to happen. At least not yet. Hopefully, if you guys become the leading nutritional organization in the world, it will, because you guys are interested in these things. But in the meantime, the people coming to you are interested in this. So you can help them get it through food, but it's really, really hard to get enough vitamin K2 through food. Vitamin K2 is made in the gut from bacteria from vitamin K1, and so it's a fermented product like natto that gives you the vitamin K2. Otherwise, you just won't have enough. So a lot of times you have to supplement. So 50 micrograms of K2 is really safe for everyone, even people on blood thinners. That study has been done with people on Plavix. I just noticed that I don't have that much time left, so I'm going to talk a little faster. Inflammation, curcumin is one way to reduce that pathway. It comes in a lot of kinds. Make sure you use an absorbable type, something that's like Mariva, where it's attached to phosphatidylcholine, or a micronized like theracumin. Next best after those two are BCM95, and then with piperine. Green tea, that's something they can put in their diet, addresses inflammation, or they can take EGCG, and that affects all kinds of oxidative stress pathways. Quercetin from onions, from apples, has anti-inflammatory benefits. Let's look at blood sugar. I think of blood sugar as kind of, because Medicare, it's one of the few things that actually is covered for nutritionists and dietitians, is for them if they have diabetes. So that's something that you guys spend a lot of time on. But if you know someone's genomics, and you have someone whose hemoglobin A1C is starting to go up, or that has early diabetes, you're going to be able to give them more specific counseling to recommend of how to reverse that. There are pathways that are incretin pathways. There are pathways that are exercise mitochondrial pathways like PPAR, ones that relate to stress, the ADR receptors, the adrenaline receptors. And all of those have different modifications. So we'll take a quick look at a few of them. The TCF7L2 is the incretin pathway. And again, PPAR and the melatonin ones are the other ones I'm going to look at. If you have variants that are known to be significant in the transcription factor 7-like-2, which affects incretin, one copy of that decreases your incretin effect by 30%. What does that mean? You can give someone IV glucose, they're going to release enough insulin, but when they eat their food, they're not going to get the signals of the incretin that then has them release enough insulin. So what can you do about that? Well, a low glycemic index diet would be the first thing, because then they don't need as much insulin. A diet that's high in fiber, high in low glycemic index foods. Whey protein. So whey protein is different than pea protein. Both proteins, all proteins, will help keep the blood sugar low. Whey protein has an additional effect of causing increased insulin release. So it's not for everyone, but if you have somebody who has less ability to release insulin because of their incretin pathways, that may be a great option for them. Especially if they want to have a smoothie or fruit in the morning, which is a fairly high carbohydrate load. Have them put some whey protein in their smoothie. Gymneta sylvestra, ginseng, also can all affect this pathway, the incretin pathway. Gymnema sylvestre is an incretin mimetic, so it's not Victoza, but not everybody needs Victoza if you catch them early. It's not quite as strong, and it can have a significant effect on people where this is their issue. There's other benefits. Gymnema sylvestre has anti-ulcer and anti-allergic benefits. It's actually also known to decrease cholesterol and affect fatty acid assimilation in the small intestines. So that's a great supplement. It's cheap. I mean, you can get Nature's Way blood sugar where the main ingredient is 250 milligrams of Gymnema sylvestre for like $7. So it's a pretty cheap thing. PPAR. How many of you guys have heard of like the PPAR drugs, right? Well, this PPAR receptor, PPAR pathways are actually mitochondrial pathways. And this is a PPAR receptor, but it's affected in mitochondrial biogenesis. So if you have your main risk in that pathway, the things that affect mitochondria are going to be what's going to help. Exercising, especially in the cold. Fasting, having a later start to breakfast. The different supplements, alpha lipoic acid, CoQ10 that you think of as mitochondrial. A low inflammatory index. There was actually a diet study done, and there's a low inflammatory index where they gave numbers as to how it affected these PPAR pathways because they have implications, some of the SNPs, in colon cancer, and they were able to show that. High fructose corn syrup is not good for this pathway. And the last one in the sugar pathway is the melatonin receptor. You don't want to release a lot of insulin at night while you're sleeping because you don't want to drop your blood sugar while you're sleeping. So people who have a variant in their melatonin receptor, they kind of hang on to their melatonin later. So they shouldn't have an early breakfast because they're not yet ready to release insulin. The last pathway I want to talk about is homocysteine. And homocysteine is that one carbon metabolism pathway. The reason I want to talk about it is you look at that pathway and it just makes you want to go like, oh my gosh, I cannot learn this. It's a lot of pieces. But I want you to think about the fact that, because the one carbon metabolism, the folate pathway, the methionine, this is where we get our purines from to make that letter T and make those different nucleotide bases for our DNA. This is where we get the methyl groups to turn on and off genes, to make our neurotransmitters. It's actually a really important pathway. So homocysteine is a byproduct of the pathway, but it's kind of an intermediate step to making glutathione because you need cysteine for glutathione for the detox. So the thing is, if you have backups in any one part of this pathway, you're going to have high homocysteine. And the question is, how can you target helping to lower homocysteine? High homocysteine is associated with increased neural tube defects, with stroke, with migraine, cognitive impairment, heart disease, macular degeneration. The list goes on and on. So it's a pretty important pathway. And so we have three ways, basically, to get rid of homocysteine. One is we can convert it back to methionine via MTR and the MTRR, and that's with B12 as a cofactor. Or we can go down the transsulfuration pathway with CBS, and then if you have a lot of variance in that CBS pathway, you're going to want to make sure they have enough B6 because that's a B6 dependent pathway. And if they don't have enough B6 and that pathway is already working poorly, it's going to work less well, and they won't be able to make their cysteine and their glutathione. But you also can have a third way where you can kind of convert it back to methionine with the BHMT pathway. That pathway is pushed by betaine, which is not just beets, also spinach and lots of other foods have it. Betaine, by the way, is also called TMG, and that's a zinc-dependent pathway. So if they have problems in that BHMT pathway with known variants that affect this homocysteine level, you want to make sure they have adequate zinc. And again, betaine is made from choline, so if they have BHMT variants, you also may need to make sure they have adequate choline. And again, there's some slides on some high betaine foods for fun. And so the potential interventions for high homocysteine depends on what's causing their high homocysteine. And it may be all three of these. And again, that allows you to counsel the patient saying, here's how you can do it with food. My dietitian has made wonderful lists of all the different foods. Come and see me if you want to know how you can get access to all of those. I had her do all the different handouts. Here's the high betaine foods. Here's the high choline foods. Here's the high methionine foods because lowering methionine can also help. And all of these different things can help you do targeted nutritional interventions as a first step and then use supplementation if necessary. I'm going to put a quick special attention to that MTHFR. The C677T, that famous MTHFR SNP, is in the riboflavin binding site of it. And in order to decrease the risks associated with MTHFR C677T, you have to have adequate riboflavin. So it's a cofactor, and it's very important for that particular SNP. So you can see why so much of medicine and health really depends on you guys. It is nutrigenomics. So genomics is a tool. I don't believe that we're going to go in and say, okay, let's address every single SNP in your genome. None of us have the knowledge to do it or the time to do it. But if you know what runs in someone's family history, if you know what symptoms they're starting to have, if you have their labs, their metabolomics, you have their, whether you have organic oats testing or whether you just have their homocysteine level from their blood, but as you start to get to know your clients, they have high blood sugar, they have obesity, you can use their genomics to help them understand themselves. And people will find it very motivating when you can say, for you, this is likely to help us address your obesity or your blood sugar or your homocysteine level. This is likely to help protect your heart. And I really firmly believe that that ounce of prevention is worth the pound of cure, and that by using genomics, we are able to do it on a personalized level. When I gave a talk about 1.5 years ago to a group of 400 family physicians, I asked them to raise their hands if they had ever used genomics in their practice. Room of 400 people, 3 people had raised their hands. It was really, really sad to me because remember, there's also pharmacogenomics in-package inserts of already many, many drugs. And so the last thing I want to say is genomics is here. As Dana said, it's already happening. And so this was the cover of the Family Practice Journal the week before I gave the talk. Buckle up. Personalized omics profiling is here to stay. You guys have met that challenge and are here so that you can accept that challenge and do it in an academic way. And so again, congratulations for preparing for the present. You're going to find as you dig into genomics, you can use it for personalized nutrition and supplementation for really just about every single disease state. But don't expect to go home on Monday and feel like you know the whole field of genomics, because it's learning a whole new field. I describe it as going from an X-ray to an MRI. You're not going to be able to go from reading an X-ray to an MRI in an hour. But just start with the things. If you have a lot of diabetes in your practice, if you have a lot of fatigue in your practice, or high homocysteines or whatever, start by learning a few things and then expand as you go along. I only have about 2 minutes. Are we going to do questions later so we don't do questions? Okay, well then I'm on time. So thank you for having me, and I'm going to next.